introduction, and now we move on and come to the next speaker. That is Clemens Falker Gieske from the University of Göttingen in Germany, and he's going to talk about a very serious problem in chickens, that is feather pecking. So please, Clemens, the floor is yours, and we are looking forward to the presentation.
Yes, thank you very much for inviting me and giving me the opportunity to present my results that I gathered over the last few years on feather pecking. And today I will try to convince you that feather pecking is a dysfunction of the GABAergic system in the brains of these chickens. So feather pecking severely impairs animal welfare and causes economic losses. In the old days, this was controlled by beak trimming, but this is now, at least in Germany, banned and not allowed anymore. It is a heritable and complex trait, but very hard to measure. And for that reason, commercial flocks are not really of good use to do research on feather pecking. Therefore, selection lines have been established to study the behavior in egg-laying chickens. Explanations for the behavior so far are that it is not an aggressive behavior, so it's rather an obsessive behavior. It's generally accepted that the dopaminergic and the serotonergic system is involved. These chickens seem to be hyperactive. And obviously, this is a redirected foraging behavior driven by the eating of feathers. The gut microbiota don't seem to be involved. So our partners in this project didn't find any hard evidence that the gut microbiota plays a role. But several groups in the past already suspected an involvement of the immune system. I will get back to that later. So we need a deeper mechanistic understanding and knowledge about the genetics behind this behavior so that we can improve it by selection. So we came up with this project. I will only focus on the parts of the project that I actually worked on. So the chickens that we analyzed are based on one founder population that you see here. And from there, two experimental populations were established. One is an F2 design over here. These were genotyped with a 60K chip, and also part of them was whole genome sequenced. We used these whole genome sequences to impute these chip genotype chickens to whole genome sequencing level to do GWAS on them. We did the same in a half-zip population that we established, but here we additionally performed RNA-seq to do differential gene expression studies between high and low pecker chickens. And finally, we wanted to do an expression quantitative trait loci study, an EQTL study. And I will briefly go into all of these aspects in the next few slides and only point out the main findings from these experiments. So by comparing high and low peckers regarding the genes that are differentially expressed in the whole brains of these animals, and we came to two conclusions in the end. So we also found an involvement of the immune system. In several gene enrichment analysis, we mostly found terms that are related to the immune system. Furthermore, we found about 40% of these differentially expressed genes to be non-coding RNAs. And that is something that has already been described in schizophrenia in humans, where in 2018 in a science paper, these RNAs were termed psychiatric non-coding RNAs. We also took a look at the light response of these animals, because light is a major trigger of feather pecking behavior. So we analyzed animals that were kept in the dark and compared them to animals that were then subjected to a light stimulus until they showed feather pecking behavior. And here you see differentially expressed genes in the brains of low pecker chickens. And as you can see here, you get a strong induction of genes to the light response and almost no downregulation of genes. You see a completely different picture if you look at the high packers. So we only saw one gene upregulated in comparison to over 400 here. And we saw about 15 genes downregulated in the high packer brains in response to light. So we took a closer look at only these genes that you see up here in red in this heat map. And as you can see, these genes that are downregulated in the high packers are mostly upregulated in the low packers. And two genes I want to point out that are especially interesting, which is here DISA1 and the GABAB receptor 2. Because what we conclude from these results is that a downregulation of DISA1 in the high packers leads to a decrease in microRNA production. And this is a link that's already been established in humans, that DISA1 further downstream controls the expression of GABA receptors in the brain. So this is something that was also found in schizophrenia studies. So this results in a high steady state levels of neuronal excitation in the high packers. And that's why we can't really induce gene expression, even with the most basic stimulus like light. So this is one conclusion we drew from this light experiment. Then I conducted several genome-wide association studies on different classes of genetic variants. On the left, you see GWAS done on single nucleotide polymorphisms in short indels. And here you see GWAS conducted on structural variants and tandem repeats. I don't want to go into many details. So here you see the top associated genes from all these GWAS analysis. And again, here we find a GABA receptor that is associated in multiple GWAS. This is the one down here. And we found a microRNA that was significant or highly associated in all of the GWASs, and predicted downstream targets here again are GABA receptors. And additionally, we found multiple genes that are also involved in human psychiatric disorders. So we combined the two types of data, the transcriptomic and the genomic data, and performed expression GWAS on over 80 genes, and produced an association weight matrix from that, and detected significant gene-gene interactions. And also here we found an interesting gene right in the center of this gene-gene interaction map that you see here, which is dystrophin, and this gene has been shown to be required for GABA-A receptor clustering. Also, we found a few genes that have been linked to schizophrenia in humans. And what's most interesting, we found two transcription factors in this analysis. One of those is termed SP6 or KLF14. And one of these, the CHTOP, contains a SMAD4 domain. I performed transcription factor binding site enrichment analysis, and we found that for this KLF14, that this deletion downstream of the gene that I found, which is associated with a differential expression of 40 genes between high and low packers, that we have a significant enrichment of transcription factor binding sites in those 40 genes. 10 of those play a role in the immune system. And in 2020, a paper in Cell was published where they showed that for the developing brain, CD4 T cells are required to for synapse development. And if there's a disturbance in the system, this could lead to behavioral abnormalities. And we think that this might also play a role in the feather pecking behavior. And this brings together the immune system part with the behavioral, the behavior that we see in those chickens. I did another EGWAS, this time not with SNPs. This time I did it with structural variants and tandem repeats. And again, I found this dystrophin gene that I just talked about, which is important for the clustering of GABA receptors. We found 3 other genes overlapping between the 2 AVMs, and we found an additional significantly associated transcription factor, which is called ETV1. I did the same thing here. I checked for transcription factor binding enrichment in the genes that are associated with this transcription factor. And again, the result was significant. Again, here we see the link to the immune system. So this ETV1 has an influence on T cell gene expression, and it also forms complexes with SMAD4. So if you remember 2 slides ago, this other transcription factor contains a SMAD4 protein motif. So we also see a link here between the 2 analyses. So we found further evidence for involvement of T cells in the neurodevelopment of the chickens. So to sum up my results, at the moment we have 2 main working models for the feather pecking behavior that might probably interact. So we have a low GABA receptor density in the neurons of the high peckers. This is probably caused by a DISO1 dysregulation, which leads to a shortage of microRNAs that regulate these GABA receptors. Furthermore, this dysfunctional dystrophin might lead to impairment of GABA receptor clustering. So low concentrations of GABA or a low responsiveness to GABA in the brain of the high peckers might cause a constant high level of excitations in the neurons of those chickens, and might explain this obsessive compulsive like behavior that we see. Then we think that the feather pecking, at least in these lines that were divergently selected for the behavior, already starts in early development caused by mutations in proximity to these 2 transcription factors, which are significantly associated with genes involved in the immune system. And we think that an early dysregulation of the T cell expression in the developing embryo might be an explanation for the development of feather pecking in chickens. Okay, that's what I wanted to conclude with, and I'm open for questions now. Thank you.
Excellent, excellent, and perfect in time. So
yeah, I have a timer here.
Excellent. So, Michelle, you want to moderate the questions, or?
Yes, there are 3 questions. So, but maybe the speaker can see them. So the first question from my colleague Elisabeth Baeza-Campon is whether you analyze the whole brain or only a part of the brain to study gene expression.
Yeah, we only did the whole brain. We discussed that while we developed the project, and nobody in the team had the necessary neurosurgeon skills to extract certain parts. So we went for the whole brain, yes.
Okay, and then it seems that this mechanism, so a second question from the same colleague, is well fixed in the brain of the hens. In another species, Muscovy ducks, there is a pecking behavior which begins at 4 weeks of age and decreases after 8 weeks of age, following the development of feathers. So how can you explain this evolution of pecking behavior with age as compared to your results in the hens?
Good question. I think what we are looking at is a pretty artificial system, because they have been selected and bred for this behavior over several generations. And I think what we see in our lines is an accumulation of several mutations that finally lead to this extreme feather pecking behavior that you don't really see naturally. So I can't say anything about age in that respect.
Mm-hmm. But at least in hens, it does not take place at the time of development of feathers. It's only in adults what you observe, or did you observe a behavior also in the young?
To be honest, I don't know that, because I wasn't involved in the observation of the chickens.
And so there is a question also from Douglas Rhodes about the other trait differences between the high and low peck lines, production trait differences, egg number, egg weight?
I think we had a master thesis about bone stability, and as far as I remember, high packer bones weren't as stable as those from the low packers. And I think there's also different in the eggshell, but to be honest, I don't know exactly now. But with the bones, there's definitely a difference.
And the last one from Hans Heinrich Thiele, that flocks which are good, well managed, do not show any pecking, or at least not all their life. So, but they have those genes that you have been identifying. So how can you explain the fact that there is no constant pecking, fortunately, in well-managed flocks?
I think it's related to what I just said 2 minutes ago, that the chickens that we are looking at were specifically selected for the behavior in order to identify genetic risk loci. And if certain flocks have problem with the behavior, we could look if the how high the allele frequencies of those mutations are that we found, and probably breed against that. But if you don't have the problem, then you don't have to think about it, right?
Thank you very much, Stefan. I think we are on time.
Thank you very much for this really interesting talk, and there are certainly some more questions.
Thank you.
But